Uh, Hello, everyone. So today we have a great talk by Leonid Peshkin. He will present us very interesting problem, which is between machine learning, statistics, and biology, pharmacology. So, who is interested in this topic, please contact Leonid or me after this talk. So, Leonid, thank you very much that you agreed to make a talk, and please start.
Mm. All right. All right. Thank you for this opportunity to present. Here's my email. I spent several months visiting Institute Curie last year, but usually I'm at the Harvard Medical School. This is going to be a short talk. We could then dive deeper in as necessary. I decided to actually start from the end here. I will describe a method that we developed many years ago, and it was co-developed by Sasha Kagan, who is here in the audience. So first, application. I'm not sure people are familiar with InQSight. InQSight is an instrument which allows to record plated cells. So I jokingly created this cartoon to illustrate what's known as a scratch assay. So in the scratch assay, you take cells, a cell line, plate those, and then make a wound, leave cells be, and then cells do what cells normally do. They divide and migrate. And so the wound is being filled in. Okay, it's an example of macro-phenotype of large-scale behavior, right? This is not, what's important is it's not a measurement, not a molecular measurement, not gene expression or protein expression measurement, but an aggregate phenotype of a more or less complex system. So you could, using this InQSight, you could record wound healing. And then here's a plot which shows relative wound density against time in hours. And what you see is that wound density equilibrates with density outside of the wound, right? The wound is closed. And so there are several plots which demonstrate for this specific cell line how the wound is being equilibrated, healed in the presence of a drug. Right? It doesn't really matter which cell line and which drug, but you see that as you increase concentration of this drug, you slow down the process. And it could be for many reasons. It doesn't matter. It could be that cell division or cell migration is impeded. What's important to notice here is how tight the error bars are. Right, so it's a very quantitative phenotype, which is hard to get, hard to obtain in biology. Right, solid numbers in biology, in my experience, are very difficult to get, and this is very repeatable phenotype. So if you add a little bit of this drug, and you impede cell migration by 10 or 15%, it is a measurable phenotype. Okay. So then, what you want to do, assuming that you start with phenomena, right? And I say this is biologist visiting a mathematician. Biologist shows up and says, I have this phenomena. I have no idea what is molecular mechanism, what is involved. Can you get me some insight? Which molecules, which genes, which pathways are involved, are controlling this phenomena, wound healing? And I apply the method I'm going to tell you about, and produce a list of genes which are responsible. So it happens here that the list is a list of kinases, and you could verify that predicted kinases are in fact important. These genes are in fact important in the process by, in this case, siRNA. All right, so you say you block the RNA of this gene, you then repeat the assay, relative scratch density, and you see that in fact blocking the expression of this kinase affects your phenotype. So that's a way to validate prediction. So this work has been done now, 10 years ago. We published a couple of papers. So if you read this QR code, you could look at the papers. One paper describes the method itself, and this is a powerful application to revealing which kinases are important in cell migration in metastasis. This method has been applied since by several groups, including us, to various other macro-phenotypes. So growth of endothelial cells is one example. Secretion of cytokines with COVID infection, and so on and so forth. So several successful applications to reverse engineer a phenotype. And let me tell you how this works. I should also say that yet another application is in progress, and maybe Sasha will have a slide or two at the end to show what he is doing with entirely new biological phenomena called anastasis to apply this method. Okay, but so how does this magic works? Right? Again, somebody shows up, tells me there's a macro-phenotype, and I come back with a laundry list of suspects, of molecules which are involved. So there is a very naive assumption illustrated by this plot that, well, there are genes, protein-coding genes, which activity of which affect pathways, which in turn control activity, lifespan, fecundity, macro-phenotype, okay? And so this is ridiculously naive, right? It's feed-forward diagram. But we sort of, with abuse of notation, use an assumption from physics that if your system is near equilibrium, if you perturb the system a little bit, then even simple models would provide some insight. So linear models would adequately reflect what's going on near equilibrium. So how do you provide a little bit of perturbation? The only way known to me to perturb biological system a little bit is through drugs. Because with drug, you control, you know which way causality flows, you control the concentration. You could just add water, you could add a tiny bit of a drug, you could add more. And so then drugs perturb activity of proteins, and so causality flows left to right. So gentle perturbations with drugs. Now, this slide is not appropriate for this audience. This is how I explain to biologists what regularized regression is, right? But let's go through this. So D1 and so on are drugs. K are kinases, activity of kinases, and P is a phenotype. So I assume that we have this matrix where for each drug, we know how this drug, which happens to be inhibitor of kinases, affects the activity of kinases K1, K2, K3, okay, in a cartoonish way. And we also measure phenotype. So in our case, we measure how rapidly a wound has been closing. We can measure how red I get in the face when I take this drug. We can measure how much of a certain substance is secreted, and so on and so forth. So then observation here is that if we forget that our perturbation was induced by the drugs, and we simply plot activity of kinase against the phenotype, then there's going to be some case with linear dependence, right? So we say, okay, it looks like K1 actually affects the phenotype because there is correlation, not K2 and K3. And in fact, this matrix here, the matrix of drug effects on activity of kinases, was available when we started 10 years ago already. You have a large number of kinases, and there are total 400 or 600 of kinases in human genome, depending on how you count kinases, against hundreds of inhibitors has been measured. Okay, it has been measured by somebody once, and so for many hundreds of drugs, you know how active certain kinases would be in the presence of different doses of those drugs. So then what you could trivially do, obviously, if you have, let's say that we've done this measurement of kinase activity in the presence, okay, let me illustrate it here. So if you ever heard of kinases, you have seen this image. This is an image which shows relationship between 600 or so kinases encoded in the human genome. This is called Kinome. And all of those protein coding genes are related. This tree was built based on sequence similarity of catalytic domain of kinases. So there are tyrosine kinases in this branch, for example. And so using this diagram, what I've done is an illustration of what happens to activity of those kinases in the presence of a drug. So one drug, another drug, third drug. Let me play this movie. As I increase the dose of a kinase, you see how much different kinase branches are affected, right? The bigger the disk, the more inhibited are the kinases. And so you will see that different drugs have different specificity. As you increase the dose, some are very dirty and rapidly inhibit everything, others remain pretty specific. Okay, so kinase inhibitors are generally dirty drugs. They were originally thought to be great cancer drugs, to be specific inhibitors, but they are not. And in a sense, this approach takes advantage of the fact that they are dirty drugs. So obviously, if in this matrix of which kinase is inhibited to what extent by which drug, we would have two identical inhibitors. Let's say I cheat. I take I take one drug and give it two different names. It would have identical lines in this matrix of inhibition. Right, so there's no information you gain by using the same exact drug twice. And if you want to select most informative kinases, you could just run principal components. And I call this principal compounds. There's a bit of a variation there, but you could select 3 or 30 most informative inhibitors from this matrix with this linear principal component kind of approach. Okay, so then this diagram of all the kinases illustrates how well we do with currently available hundreds of inhibitors. So what can you ask about all of the kinases? First of all, you want to know are all of the kinases identifiable? Something I should have said in the beginning, I apologize, is why can we actually focus only on kinases, right? I told you we're reverse engineering phenotypes, and we want to find which molecules and which genes are responsible. There are 22,000 protein coding genes and only 600 kinases. Why kinases? First, because we have this wonderful dirty drugs which allow for this approach. Second, because many pathways have several kinases as part of the pathway. So if you identify the kinase, then guilt by association, you can also identify through a kinase, you can identify which pathway is involved. Okay, so we want to ask, well, are all 600 or so kinases inhibitable by our set of drugs? And so this slide illustrates the answer. Red means kinases which more or less specifically are inhibited and inhibitable. Gray are not measured at all in that weight matrix. And blue is what we call dark kinome. Those are not distinguishable with the current assay. So we're doing pretty well with the pink identifiable kinases. So going back to our diagram, in order to identify guilty kinases, we apply textbook machine learning approach, which is regularized regression. So it should be familiar to people in the audience, right? This is just plain vanilla linear regression penalized for complexity for the number of terms. We regress the measured phenotype to the activity of the kinases, and hopefully there is an optimum which gives us the answer. So let me now give you step-by-step method which we use to summarize what we're talking about, and then I will mention open challenges here, which I hope somebody in the audience will come and help us solve. So the first step is we select the broad, we select a broad set of drugs and drug targets and obtain this interaction map, right? So the interaction map is only obtained once. It is expensive, long step, right? It costs about $1 million to run all of the pairwise assays of kinase inhibitor by kinase. You do it once and you have this giant matrix of hundreds of inhibitors by hundreds of kinases. So then you set up continuous phenotypic assay to characterize the effect. So it could be wound healing, cell growth, apoptosis, and so on and so forth. Okay, so then you select small representative subset. Those are our principal compounds, which are spanning the set of inhibition. Perform and measure the phenotype in the presence of these principal compounds. So then you perform data analysis of regularized regression to create this naive linear model. You can apply the model to untested drugs. It has predictive power, and identify possible cocktail which would specifically or as specifically as possible inhibit kinases of interest. Okay, so this is the workflow. Maybe I should no, let me carry on and then we could get to questions. Okay, so where do we need help? This whole framework is extremely naive. I'm shocked that it works as well as it does. So first of all, notice that we actually don't work with linear functions. We work with a sigmoid space. So this is activity of a kinase, of a given kinase, in the presence of inhibitor. And of course, it starts with full activity. It goes down to zero eventually for most cases. And it's a sigmoid. We do not take this into account, but we should. There is also a sigmoid where we map activity of the kinase onto phenotype itself, right? So the change in activity of kinases responsible for phenotype should also have sigmoidal shape. Now, the method of regularized regression essentially treats every kinase, every variable, as an independent variable, which they're not. I just showed you that it's a tree. Some are very close relatives, and some are very distant, and similarity measured between kinases should be taken into account. All right, that's another question. Now, we identify kinases, but what do kinases do? Kinases phosphorylate sites on proteins. It would be very helpful to identify not just the kinase involved, but take the next step and identify which phosphosite is a target responsible for a particular phenotype. And so there are a couple of papers, quite recent, with an atlas of substrate specificities for kinases, right? So this specificity is defined as another matrix. So somehow taking advantage of both having this matrix and this matrix in order to identify phosphosites is also of great interest. Next challenge. What I suggested in my workflow is we select a set of principal compounds, be it 20 or 30 compounds. We apply those compounds to test the phenotype, and then we go home and do the analysis. But it doesn't have to be this way. Obviously, there is some way to optimally select the encoding. And many people mentioned to me, oh, this is a solved problem. There is a theory of optimal control from the 60s. There is a theory of optimal information encoding, but no one was able to actually map this problem to a solution yet, right? So the problem is, let's say your experiments are very expensive and you want to start by testing one drug, one kinase inhibitor. So looking at the matrix of inhibitors, you should select the most informative one, providing most information. You do this experiment, possibly with several doses, and by the way in which kinase inhibitor affects phenotype at multiple doses obviously is also related, okay? We don't know how to encode this mathematically. So you try this one drug, you come back and you say this drug has no effect. Provided this information, then in sequential manner, you can pick the next one, which is the next most informative drug, and so on and so forth. So what is the correct decision tree of selecting drugs which would get you to the answer in the fastest possible way? Okay, there is a dual problem here, which is, so far I've been talking to you about a suspect, about the kinase or a few kinases responsible. Well, it doesn't have to be the case that we identify the kinase, because if we know nothing, right, if we have an important biological phenomenon and we know nothing about molecular mechanism, even if you point me to a family of kinases, it's already a gain. If you just tell me, well, I don't know which one, but the guilty kinase is somewhere in the branch of tyrosine kinases, that's good. If you narrow it down even more to a sub-branch without pointing me to a specific one, that's good, that's helpful. And so again, if I give you a budget of drugs, if I tell you I can run 10 experiments, looking at the matrix of inhibitions, you should be able to select 10 best, most informative drugs to narrow down which branch is important. And again, I'm imagining some sort of maybe Bayesian formulation, where you would formulate a prior on the tree, and I do not know how to work with Bayesian inference on trees. You could start with some sort of uniform prior and then update given every drug, and evolve your likelihoods on the tree in order to provide the distribution at any point and summarize it. Okay, right, so I would be helpful for any leads to how to properly think about this. Okay, I think that's enough said. I've been talking for 35 minutes. So let me stop here. I think it makes sense to maybe stop recording and then go to questions. Okay, and also if Sasha wants to add something or show a slide or two, that would also be a good moment. Sasha, do you want to be recorded or?
I don't have slides, but I'm ready to answer technical
Okay,
questions.
let's stop recording then and go to questions.